So the second part of protein synthesis is translation. And the idea for this is that it happens in the cytoplasm. Okay, this is one of the key differences between transcription and translation. And overall, so in transcription, you're producing an mRNA molecule. That's the point of it, is to make a genetic copy so that you can produce a protein. And so here, this is what you're doing: is you start with the mRNA, and you're actually going to produce the protein itself, whatever that is that the body needs. All right. So you start with the mRNA. I'm going to pause it here and draw my mRNA because it might take a while otherwise. So this is my mRNA molecule. And so the first thing that happens is in the cytoplasm, the mRNA associates with the ribosome. All right, so, or you could say the ribosome binds to the mRNA, but as long as you have the idea that these two are interacting with each other, you're okay. All right, so the mRNA associates with the ribosome. And basically what that does is that it sets it up, right? And remember, the idea is that the mRNA is read 3 bases at a time. Okay, that's a codon. You want to mention that. So the idea is that the first thing that happens is a tRNA enters the ribosome. The ribosome would be like this enormous thing here. I'm not going to draw it because then I don't have room to write, but it would be this kind of enormous molecule that surrounds the mRNA. Okay, and so you have a tRNA, and it's always carrying a specific amino acid. This is an important thing that you have to mention. The idea that it's not just any old amino acid. It's not just an amino acid. It's a specific amino acid. So that enters the ribosome. And the idea, of course, is that the codon, right? So this is a single codon on the mRNA. So the tRNA has an anticodon, which, God help me, I don't want to try to draw tRNA here, but the idea is the anticodon would be what GUA here. Okay. So the anticodon on the tRNA is going to, again, so it's going to match, right? This is still complementary base pairing. You can mention that here if you want. And I would mention that this is a triplet codon, right? The idea that there are 3 bases involved in this is always worth mentioning. So it binds to the triplet codon on the mRNA, and again, mention how it's happening. So this is via the complementary base pairing rule and the bonds that are formed. And the bonds that are formed, again, with all of these complementary base pairing, these are hydrogen bonds. Binds triplet codon. Okay. And then the idea, whoops, that's number 4. So the idea is that then you have a second tRNA enters. Okay. And so the idea is this first tRNA, which I'll just draw as a cartoon. So that's your amino acid there, is held in place by the ribosome, and you have a second tRNA enters. And also matches up. Okay. And the idea is that again, always name the bonds, right? So a peptide bond is, sorry guys, is formed between the adjacent, or you could say between the first and second amino acids. Right, so if you have another one here, let's see, this is going to be C, C, and G. And so so these are your 2 amino acids, and the idea is that you're forming a peptide bond here. All right. Peptides between the adjacent amino acids. And then, of course, sorry, you have, so the first tRNA leaves, another one enters. You could say, minus, it doesn't have an amino acid anymore. So sans amino acid, if you want to be fancy about it. Another enters, and the idea is that the process continues until stop codon is reached. So, and so how would I phrase this? I would say, so the mRNA continues to be read 3 bases or a single codon at a time. Until a stop codon is reached. And a stop codon is basically a codon for which there is no tRNA, or the and so if you don't have a tRNA they can bind there, then the whole ribosome falls off and is done. Now, the other thing I didn't mention, and I apologize for this, is here where the tRNA is carried, it enters the ribosome. So here between these two, the mRNA associates with the ribosome, and then the idea is that the ribosome actually scans the mRNA until it encounters a start codon. Okay, so you could add here until a start codon is reached. Usually there is a point in mark schemes for the idea of start and stop codons, so it's always worth mentioning, right? Sorry, just add that onto step 1. Okay. And the idea is that what this does is it creates a polypeptide chain, which then, of course, upon release from the ribosome will fold into its final tertiary structure. Okay, so these are this one and the other one together kind of hopefully gives you an overview of the protein synthesis process. But the other thing that's important to remember when you're looking at these two protein synthesis ideas is that you want to make sure that you understand what's going on, so that if they ask you about mutations, you are able to talk about what is actually changing, right? So the idea is that if you have a change, if you have a mutation in the gene on the DNA, what, they often say like, well, how is that going to affect the final protein? Okay. And so you need to be able to walk through, you need to be able to say, well, if there's a mutation, if there's a change in the base, right? That's what a mutation is. It's a change in the sequence of bases. So if you change the sequence of bases on the DNA, you're going to get a change on the mRNA molecule that's produced. And if you get a change on the mRNA molecule, then you're going to get a change on the codon or on the amino acid that is this, because a change in the codon would mean a change in the anticodon, and a change in the anticodon would mean that the tRNA could be carrying a different amino acid, which means that now you've altered the primary sequence or the primary structure of the protein, which then means, of course, you could alter the tertiary structure. So you have to be able to connect all of these things in your mind, all right? And don't forget to apply it. So this is the overall process, right? But if they ask about a specific gene, then rather than just saying, that this is happening, you would say that the nucleotides match with the exposed bases for the gene, whatever it is. Okay, and the same thing here. The idea is that you would talk about the mRNA needs to be a copy of the specific gene that they are asking about. So don't forget to apply this information to whatever specifics they are giving you.